let me introduce the speaker of today, Jonathan Heddle. Jonathan completed his PhD in biochemistry from the University of Leicester in UK. Then he moved to Yokohama City University, where he carried out research in structural biology laboratory. This was followed by the two PI positions, including Tokyo Institute of Technology. He moved back to Europe, and his current position is at Malopolska Centre of Biotechnology of Jagiellonian University, and this place is, Jonathan is present since 2015. He leads the group laboratory called Bionanoscience and Biochemistry Laboratory. And this laboratory is interested in understanding and building artificial and natural bionano machines. This concerns a wide range of projects and materials, including proteins and nucleic acid structures, such as DNA nanostructures and artificial protein cages. Jonathan Heddle hopes to continue to advance both basic development and application of these structures in the future. So this is the short introduction of our speaker. I would like to ask Jonathan to activate microphone and camera. Hello.
Hello.
Nice to see you.
And you too.
And yes, I can listen you perfect and hear you perfect. If you can share your presentation, you can start your lecture.
Okay, I hope you can all see that.
Yeah, it's perfect. It's
great.
Perfect.
So first of all, thank you, Adam, and the Society for the opportunity to speak. It's a great honor to be able to talk to you today about some of our work. The title of today's talk is Designed Protein Nanostructures, and it was funded, the majority of which is funded by funding bodies in Poland, NCN and FNP. Most important thing, of course, to do is to acknowledge the people who actually did the work. I'm just the head of the lab. We've got a great group of researchers, too many, in fact, to list all here in the time we have. But they're photographed here. It's a little bit out of date, this photo, but if you go to our lab page, you can see more of the people and the work they're doing. We also have a lot of collaborators. Protein cage work, which is mostly what I'll talk to you about today, are listed here. I probably missed someone out, in which case I apologize if I have. And I will briefly mention the Origami work, which we also have some excellent collaborators on. So what are we actually doing? Well, as mentioned, we're at the Malopolska Centre of Biotechnology. It's a fairly new institute within Jagiellonian University. For those of you who might be listening and viewing from outside of Poland, it's in Krakow in southern Poland, which is a very nice city. Please visit if you get the chance. And we are doing quite well. There's some of our recent publications are listed here, and we're always looking for good PhD and postdocs. Our contact details are here. So, what do we do in the lab? So, as the name of our lab suggests, we're interested in bionano machines. For us, what this means is natural bionano machines, i.e. enzymes, for example, and we're particularly interested in one enzyme, DNA gyrase, which is most well known as a bacterial enzyme and a very important target for antibacterial drugs. And there we're interested in protein and peptide inhibitors of this enzyme. And we're also interested in gyrases, these molecular machines, which are found in organisms other than bacteria, so particularly Plasmodium, the causative agent of malaria. And then we have our interest in artificial nanomachines, so biological structures or structures made from biological material which are not natural. So in this case, for us, this means DNA structures, DNA origami. And we have a couple of projects here based on drug delivery systems and membrane interactions. and protein structures. So what I'm going to particularly mention today is this red highlighted thing here. This is protein cages. But I just wanted to take a short opportunity to very briefly mention the other topics. So we work on, as I mentioned, we work on a protein, an enzyme called DNA gyrase, which carries out, it's really interesting from a, if you like, nanomachine point of view, because it carries out a very complicated reaction. It has a lot of, I guess you could call them moving parts. So it does a job of supercoiling DNA, negatively supercoiling DNA. And the mechanism is shown here. I don't want to go into it in any detail, simply to say that we've been looking at peptide inhibitors which are able to somehow enter this enzyme, the enzyme structure shown here, and stop it from working through the fact that they mimic DNA. So it's actually quite interesting. You have a protein which mimics DNA in order to affect a DNA binding enzyme. So this is quite interesting work because of the fact that it's a machine, and understanding how natural machines work can help us build artificial ones, but also because of the antibacterial potential. And we just published a paper on this in NAR recently. So continuing with a brief overview of other topics in our lab, we work on DNA origami. You may be aware this this technique is a way whereby you can take a long strand of DNA, a single strand of DNA called a scaffold strand, and by cutting up the complementary strand into small pieces, which are complementary to distal parts of the strand, you can staple, as the name suggests, staple together the scaffold strand to make any arbitrary shape. So this is the original work here in Nature back in 2006 by Rothman, showing a host of different 2D structures. This technique has really developed very quickly. And you can make now 3D structures. And it was some work we did with our colleagues in Hong Kong relatively recently, where we made a box which could be opened by LDH from Plasmodium falciparum. So you think of it as a protein which is indicative of the presence of the malarial parasite. So these are two areas which we are very interested in: natural nanomachines and also making structures and machines artificially from DNA. But it's the protein work I'm going to concentrate on now for the rest of today's presentation. So in particular, artificial protein cages. So these are hollow protein spheres, in many ways similar to virus capsids. Of course, viruses do a very good job of delivering their genetic cargo to cells. So there is a lot of interest in making artificial versions of protein cages. And they have three modifiable surfaces. So there's an inner surface, an inner cavity, which is useful to be able to modify, because then you could perhaps capture cargoes of your choice for example, drug delivery systems. There's an outer surface, of course, which can be modified and clearly has potential, therefore, to be used as antigen presentation, so as an artificial vaccine, or for targeting agents. So if you're looking at drug delivery systems. And then there's the interface between the proteins that make up a cage. So as you may know, you need to have many copies of the same proteins to make a cage. And so these proteins have interfaces where they stick to each other, and to be able to modify those interfaces can affect the structure and the properties of the cage, such as, for example, under the conditions under which it opens up or breaks apart. And there are many other potential uses which are listed here. So this idea that protein cages can be made artificially is not new. So as far as I know, it was suggested back in 2001 by Padilla et al. At Todd Yates's group. So the idea here is shown in this cartoon from that paper, where imagine you have a protein dimer and a protein trimer, shown here in green and red, and you can see the interfaces between the monomers of these proteins. And now imagine you fused one monomer of each protein together. So here is the structures of the actual proteins involved. And here you can see there's a connector in blue, shown here as a cartoon. And you can see that the interfaces are left unaffected. They're still open. So you can imagine that the dimer still forms a dimer, the trimer still forms a trimer, and you might get some kind of array like this, shown a two-dimensional array, or depending on if the angle is right between the monomers, you might get some, in this case, a cube structure like this. And this was essentially shown to work later in a paper from the same group in Science, where they solved the crystal structure of such a tetrahedral cage. A little bit more recently, this concept has been developed further, the idea of artificial protein cages, particularly by groups at University of Washington, especially the King and Baker groups. So this takes a computational approach. The advantage here is that it doesn't require pre-existing interfaces between proteins. So imagine you want to make, let's say, a cube. Here are the threefold axes of a cube. So you can think like the corners, right, where three faces meet. And you could imagine taking 8 trimeric proteins like this green one and sliding them down these axes until they hit each other. Now, if you're using a protein, so you have 8 copies of this trimeric protein, if this protein has no pre-existing propensity to bind to itself, then just by mixing these proteins together, you won't get a cube. But what the techniques developed by these labs allows you to do is to redesign the interface between the proteins. So you have new side chains, which will give you the required protein-protein interactions. So salt bridges, H bonds, hydrophobic packing interactions, so that you do get spontaneous cage formation. So this is quite a revolutionary idea, and it's been extremely successful. So here is a paper from the group showing structures of 10 different proteins here, and it was on the cover of Science a few years ago. So as you'll see, we've been interested in whether we could do something a little bit different, because obviously these groups are successful, and we don't think we can really match them directly with what they do, but we can look at questions like different geometries that might be available rather than Platonic solids, and programmability. I wrote here switchable assembly, so being able to control the conditions in which cages assemble, or importantly disassemble. And related to that is to look at different ways of connecting the proteins in a cage together, such that these difficult to control protein-protein interactions perhaps could be replaced with more easily designable interactions. So again, going back to the work of others, in particular Baker and King groups, this basic idea or fundamental idea of an artificial cage is now going quite swiftly towards applications, and perhaps not surprisingly, vaccines is one application. So this is a recent paper in Cell where they showed an icosahedral cage made from 2 proteins, so a trimer shown here in gray and a pentamer shown here in orange. And this is displaying a spike protein from respiratory virus, RSV. And as you're probably now very familiar with, many of these viruses have an antigen called a spike, which sticks out, and these are typically trimeric. So here you can fuse, as a genetic fusion, you can produce the gray protein fused to a monomer of spike, and then as both gray and blue are trimers, they will form nicely a fully formed spike protein. And then by mixing the 2 proteins together, you get assembly into a cage. So this will display 20 copies of the trimeric spike because there are 20 vertices on icosahedron. Similarly, a similar approach was done by the same group to show ENF-like protein spikes. This is relevant to an HIV vaccine. This was in Nature Communications in 2019. And then even more recently, again, the same groups have shown, using the same technique and the same protein cage. This is hemagglutinin, so an antigen from influenza. And of course, as you're probably familiar, influenza has lots of different, it's changing all the time. And here are some different spikes of different variants, and all of which were made and displayed on the cage. And even a mosaic was made, which has a mixture of different spikes. And also last year, this paper was published again in Cell, showing similar approach, but this time the spike, the trimeric spike, is from SARS-CoV-2. So there's been a lot of rapid progress in artificial cages, and particularly in the vaccine field. So as I mentioned, we're trying to do something a little bit different. We use a building block called TRAP. It's a species of it's a protein found in species of Geobacillus. It's an 11-membered ring, so it's made from 11 identical proteins shown here in different colors. And we've used this quite extensively for many years. So we previously modified it to make a protein nanotube, a self-assembled protein nanotube, where the rings stack on top of each other in this kind of orientation. In nature, this protein is involved in controlling the synthesis of tryptophan. It actually binds tryptophan and also binds single-stranded RNA, but this is to some extent not relevant to our work. What we found was that we could mutate TRAP, so a position 35, which is a prominent lysine around the outside of the ring, to and shown here in yellow, to cysteines. There's no other cysteines in this protein. And we found that when we added it to a source of gold-1, so we added it, we mixed it initially with gold nanoparticles, 1.4 nanometer gold nanoparticles, and we found that we could form a protein cage, which I'll show you about in a moment. And perhaps not surprising, there would be some reaction. In fact, that's what we were expecting, given that cysteine, sulfur binds strongly to gold. But the actual structure is quite curious. So these are our very first results. This is a negative stain transmission electron microscopy. And so this is the wild type protein, TRAP rings. If you look carefully, you can see, I think you can just about convince yourselves of small donut shapes here. And this is when you add nanogold. There's not really any significant change, perhaps of aggregation, but you can still see many ring proteins. Now, when you have the mutant, so this is with the cysteine, without nanogold, there's no change compared to wild type. You get these nice rings. But when you add nanogold, you see very interesting change. So you can't, in this particular image at least, you can't see any rings anymore. And you can see large structures of two sizes, in fact, about 21 nanometers and about 15 nanometers. And so these are quantized. I mean, this is not some kind of aggregation or continuum of structures, sorry. So something ordered is happening here. And these are reminiscent of virus structures, although, of course, in a negative stain TEM, you can't be sure that you're seeing a 3D spherical structure. I mean, these could be disks, for example. So this was a new discovery, and so we had a lot of very basic questions to ask. We wanted to ask, is this structure stable, for example? What are its physical-chemical properties? So we were able to challenge it with various conditions which you might expect to irreversibly break apart the protein. So this is a native page gel. So you can run the cage on a native page gel, and it runs as a single band at about a megadalton in size. Its actual size is 2.2 megadaltons, but on the gel it runs like this. And you can treat it with different pHs before you run it on a gel, and it doesn't seem to have an effect. It certainly doesn't irreversibly disassemble. And if you then, after treating at different pHs, you put it on a TEM grid, you can see very nice intact cages. So they seem to be highly resistant to pH changes. Temperature, also highly resistant. In fact, it seems to last several hours at close to boiling temperatures. This is again after 3 hours at 95 degrees C, seems to be unaffected. similarly for urea, high concentrations of urea, guanidine, SDS. So these is extremely stable. So there was another basic question, which I'll come back to towards the end, but I want to mention it now, which is that how can 11-sided shape fit together to form a sphere, you know, or a apparently regular-faced convex polyhedron, right? So there are only a limited number of such convex shapes. So the 6 platonic solids, 13 Archimedean solids, all of which are shown here, and 92 Johnson solids. And none of these include solids made from 11-sided shape. So this is a bit of a conundrum. And I will come back to answer that question at the end. Originally, we thought that, well, perhaps what happens is somehow during the interaction with gold, these rings disassemble into the constituent monomers, and okay, to maybe a rough approximation, they're sort of pieces of a pie, right? Because they're rings, and maybe we can think of them as vaguely triangular. And once you have a triangle, of course, you can build just about any convex polyhedron. So our first evidence that this might not be true came with high-speed AFM. So we were if we're very lucky, you can, and you have a good AFM session with a good tip and a good AFM, you get these kind of images of single cages, and with a lot of filtering, you could get images which suggested that annular structures were still present. So suggesting that the ring didn't break apart, that these structures were in fact made from whole rings, which didn't really answer the geometrical puzzle that we had. And the other question, of course, as well as the structure and the properties, is sort of a chemical question. What is the gold doing here? And in the end, of course, many of these questions can be answered just by getting a high resolution structure, which is eventually what we did. So this slide summarizes many years of work. So this is the starting structure, the ring. Here the positions at 35 shown in red rather than yellow. This is the same view from the side. And all we do, just to remind you, to make our cages, we simply mix in aqueous buffer at room temperature with a source of gold 1. Now, this is the source of gold 1 we use. So over the years, several years of work, we discovered that we didn't need to mix the protein with gold nanoparticles. Gold nanoparticles, it would seem simply acting as a source of gold 1. So now we can do a much simpler reaction by adding this source of gold 1. We mix them together, and we get the cages, and this is the cryo-EM. So we use cryo-EM to determine the structure. This is the cryo-EM densities. If you slice through the middle, you can find the cavity size, so that's 16 nanometers. So I should mention everything I'm telling you about now is for these so-called larger cages that we identified early on. So the whole thing's about 21 nanometers in diameter, so it's the size of a small virus. And how does it all fit together? So this is interesting. So here is the crystal structures modeled into the electron densities. And you can see, if you look at this orange ring, you can see each ring is surrounded by 5 neighbors. That's the same for all of the rings. There are 24 rings in total. And if you zoom in, you can see there are some bridges of density between the rings between monomers. Here's one monomer, here's another monomer, here's a second monomer, and here's a second monomer. And so if you go back to this, you see there are 5 neighbors. Each of the neighbors have 2 monomers attaching to 2 monomers. So in other words, this orange ring has 10 connections, and there's 1 monomer here which is unsatisfied, which is why you have these 4-fold holes, these stack holes, of which there are 6. And if you zoom in even further, you can see some density here which models nicely a gold 1 ion. So it looks like the gold is acting to form a coordinate bond between the sulfurs of the 2 cysteines. And interestingly, there was very little, if any, protein-protein interactions between these rings. So that makes this protein assembly quite unusual compared to natural assemblies like viruses or even other artificial assemblies. It implies that because you don't have to worry about other protein-protein interactions, which can be quite complex once you start changing them, it can be difficult to design and also to predict the effects of changes. If you only have one bond, you don't really have to worry about that. In principle, you might expect you could replace gold with something else. And if you could, you would be able to control the properties of the cage. So you could make actually programmable matter. I mean, you could make a protein cage whose disassembly characteristics, for example, you could program. So this is shown here, actually. This is high-speed AFM of one of these cages. And as I told you, they're highly stable. But if you add reducing agent, you'll see what happens. These cages begin to distort, and then they open up like this. And this is confirmed in the gel, so the same gels as before, but now you can see these bands corresponding to cages break apart. I mean, the bands disappear because cages are breaking apart in the presence of relatively mild concentration of reducing agent, including glutathione cellular reducing agent. So this is quite interesting to us, and because looking a long way down the line, if you could put therapeutic cargoes in there and get them to the cytoplasm of our cells, which is of course reducing, you might expect to have a very, very stable delivery system, but which then readily breaks apart in cells. We did some other work. So we looked at the possibility of replacing gold with other metals. So we showed that we could use mercury. Sorry, I think that's a mistake. It should be mercury 2, which also forms linear coordinate bonds with gold. And we found, in fact, this is the characteristic band for the cage. We found that works. And here are the result. Here's transmission electron microscopy of those cages. So they're mercury cages. We also found that we could change the position of the cysteine, and therefore you might expect that will change the angle between the rings, and therefore you would change the size and/or geometry. We got very nice cages, and we're now in the middle. Well, actually, we're almost finalizing now the structures of these cages, which are quite interesting, and I can't say much more about them at the moment. We also wanted to do exactly what I told you, to show that these cages are programmable. So can you play around with the connections between the rings such that you can then you can design and control the conditions in which the cages break apart. Breaking apart is particularly useful because, of course, of drug delivery and other potential uses. So we looked at this with a very simple programmability at first to see if we could make non-cleavable versions and a cleavable or reducible version. So truly reducible rather than what I showed you, which is probably to get into the technicalities, probably an etching reaction with the gold. So can we make non-reducible and reducible, or non-cleavable and cleavable cages? So I don't know if you can see this, but we can. So we can make so-called unbreakable trap cages. So these are cages. Here are the familiar gels, just again showing you they're pretty stable. So at a range of pHs and a range of temperatures. But now they don't break apart in the presence of reducing agents. But we made one which does break apart in reducing agents, and this is true reduction, not an etching reaction with gold. So it was successful. We're also in progress now. We're making ones which break apart in the presence of light. So I can't go into the details of that either, but here is before and after light, and it seems to work. And we're solving the crystal not the crystal structures. We're determining the cryo-EM structures. This is just an imaginary cartoon, so don't take any notice of these structures. We can also fill the cage. Again, unfortunately, not published yet. I can't go into the details except to show you some images. These are unfilled cages. These are cages filled with protein cargoes, and maybe, for example, if you see here, you can see little dots inside. Actually, this is even more cargo, and this is so much cargo the cages are beginning to not form correctly. And we can also decorate the exterior. Also can't go into the details here, but here is a cage undecorated. Here is one decorated. You can see lots of bumps on the outside, and this is of course SARS-CoV-2, a totally different kind of particle. It's a membrane. It has a membrane, and of course it's much bigger than our structures, but has the same kind of structures on the outside. So just to come back to the geometry question in the last few minutes, how does an 11-sided shape form a apparently regular convex polyhedron? So interestingly, if you join together the centers of each of the rings in the cage, you get this underlying structure shown here in black, which is actually a snub cube. So, sorry. So what we can do is let's snub cube and make the dual of a snub cube. So the dual is when you make the vertices of a shape into the faces and the faces into the vertices. So this is called, you get this white shape. It's called a pentagonal icosahedron. The name is perhaps not important, but this is a 24 face. This is a completely regular allowed shape. So you have 24 pentagonal faces shown here in white. And you can model in an abstract way, our protein ring. So, but as just drawing it as a circle with 11 points of contact, and you can see this where the points line up. This would be like where our gold would be connecting them together, and of course you have this fourfold hole here. So it seems to form very nicely, right? So what's the problem? Well, the problem is it's almost perfect, and the key point is almost. So if you're to try to draw this schematically, you draw a hendecagon, 11-sided shape inside a pentagon. But then if you zoomed in, you'd see that this line, this hendecagon actually doesn't quite fit inside. In other words, there is a small error. And the trick here is that the error is simply so small that from a large biological molecule perspective, it's really irrelevant, right? Biological molecules are flexible. So while it is in fact mathematically impossible, in reality you can get something so close to regular that you can't even see the difference. So this is just to remind you a snub cube and a pentagonal icosahedron. They're just duals of each other. Another interesting point which you maybe didn't pick up on is that snub cubes and their duals, so this is the pentagonal icosahedron again, are chiral. So you have left and right-handed versions. And so actually our protein cages are also chiral. So you can imagine that if you look at this is our cage made from 24 rings. So 12 are shown in blue and 12 are shown in red. And if you look at this ring highlighted in yellow, if you imagine pulling apart the two hemispheres, rotating and then putting it back together, you've actually gone from left-handed to right-handed forms. And just interestingly, whenever you get into sort of mathematical questions, which is obviously not my area of expertise, but you usually find somebody's got there before you. Some more mathematically inclined people have got there before you, years, if not in some cases centuries before. So, I mean, there was a paper back in 2004 which asked this question about packing of spheres on a circle, and for 24 circles you get this structure which of course our structure. So even previously, similar for n equals 24, similar results have been found, or the same result have been found. So this is, if you imagine, you could sort of imagine this as having a sphere and circular stickers which is stuck on the surface of the sphere, and how best to pack those circles. It's exactly the structure that we have. So clearly this is some quite a low energy or preferred arrangement of matter. So just about to end there. In the future, what we'll be doing, we're going to continue, of course, our basic research, looking at different shapes, different proteins, different cargos, different ways of programming these cages. And of course, we're interested in applying our research possibly to therapeutics and vaccines. So I'll thank you there. We do have positions for protein cage research coming soon, PhD positions. So please check out or contact me if you want to know more. And I will end there.
Thanks a lot, Jonathan. Thanks a lot. It was very interesting, not only because of the, let's say, scientific content, but also the aesthetic content of your presentation. It was great. So now let's try to ask the questions. As I told you, to ask the question, you have to write the question in Q&A form. That's on the bottom of your Zoom program, and then Jonathan can answer your questions. waiting for questions, let me take advantage of being host, and I will ask you the first question. Tell me about the context of the cages, protein cages, and the membranes. Is there a possibility that protein cage can diffuse through the biological membrane, or they are hydrophilic by definition?
Yeah, so I mean, of course, I can't speak for all protein cages. In our case, these are quite hydrophilic. I mean, they're highly water soluble. So without modification, you wouldn't expect them to be able to sort of independently penetrate the membrane. Of course, it all depends on cell types and also particle size. So it's quite difficult to predict, as far as I know. Actually, sometimes even that hydrophilic protein might be taken up by cells actively. So taken up into an endosome. So these are the kind of questions which often have to be asked experimentally.
Right. Mm-hmm. We have the first questions on the Q&A, and let me read you. The first is the question starting with the compliment, nice talk. Mm-hmm. If you consider these cages to be delivered system for drugs, do you expect them to trigger immune response and being destroyed before getting to cytoplasm of target cells?
That's a very good question, and that of course is always a concern with, well, I guess pretty much any foreign material that you might be injecting into a person. But not all proteins are equal. So some proteins are much more immunogenic than others. And also the advantage of, or I think one of the advantages of an artificial cage system is that they can be readily modified. So again, it comes back to, I guess, experimental evidence. You have to try, and then you have to modify. If you're talking about applied, it's kind of an engineering question. So you can imagine you might have to passivate surfaces of protein cages. But it's a very good question. It may be that the majority of protein cages will be too immunogenic to have for repeated administration.
Okay, the next question on our Q&A form. Which technology is more promising, protein cages or dendrimers? Could you please comment?
Well, I know very little about dendrimers, but I mean, they're different technologies, right? So so protein technology has the advantage that you can modify easily using genetic recombinant technology. So you can fuse other peptides and proteins to it, for example. It also and also, I think in most cases, from what I know, the protein cages such as ours, at least, which are 20 nanometers in diameter, are going to be considerably smaller than most dendrimers. So it probably depend on which disease you're wanting to treat and all those kind of details.
Okay, and the next question, including the compliment. Thank you for excellent lecture. We need some kind of the package to put inside and transfer drugs to place where, for example, in cancer. Could this be possible to be used in this use of these cages? So the context of cages and cancer.
Yes, well, I mean, yes, I mean, it's theoretically possible. You can put things inside protein cages are hollow, so they can potentially carry therapeutics, and they're modifiable, so you could potentially modify them to target. So I think the answer would probably be in principle, but you'd have to, coming back to the question about immunogenicity, you'd have to test and see.
Right. And the next question, were the spheres tested in environment containing proteases?
That's a good question. We have done some limited tests in, let's say, cell culture conditions, and they seem to be stable. We haven't done a very comprehensive protease tests now yet, although that is something we're planning to do. But actually, because they're so highly stable and they don't have much of an exposed area, actually, we're fairly sure they'll be pretty resistant to proteases.
Right. And the next question, can these protein circles can be replaced by torus-like dsDNA?
That's a nice question. So this is more of a, so this comes to our other area of DNA, like origamis, for example. Yeah, I think that there's a lot of work going on in DNA origami where you can make hierarchical systems. So you DNA origami shape and use multiples of those shapes to add together. So you would need to make something like DNA origami, not just a plasmid-like DNA, because it would be too floppy, I think. But DNA origami, obviously, it's a much more rigid structure. And I think if you as long as you have your connections, I think theoretically that could be done. Yeah.
The next question, a bit technical, but I think is this cages easy to producing?
Yes. So the basic protein, the ring. So one of the advantages of these triggable systems is that you can produce these very large structures, in our case 2.2 megadaltons, as just the monomer cage, which is less than 100 kilodaltons, the single cage. In fact, the monomer is 75 amino acids, but it assembles in the cells to make the ring. But then you can purify that ring, and that's highly soluble. I mean, it's a typical, lovely bacterial protein, as you often find, that you can get, even in laboratory conditions, you can get 100 mg per liter without too much trouble. And then you just purify it, standard purification perhaps for protein, and then you simply mix it with a source of gold. So as I said, you have a standard tris buffer, no high temperatures, no high energy, nothing special, and you mix it with the gold and you get assembly. And at least we found that yield is surprisingly high. So I think the best we've ever got is something like 80% cages formed. So yeah, it is actually relatively straightforward. You then have to purify the cages, of course, to get rid of any excess gold, but that's just done by size exclusion approaches. So yeah, it's quite easy.
Now the similar questions, again, with the compliment, thank you for a great talk. How can cages be loaded with cargos? Is there a single procedure, or it changes depending on the cage?
Well, unfortunately, I can't say too much about that because of we have some restrictions on what I'm allowed to say, but they can be loaded with cargos, as I said, and there is more than one approach. I'm afraid I cannot say any more than that. You'll find we will have a publication on that relatively soon. So,
and let me ask there is still question. What is the cut-off of the cage wall? What is the minimal size of the molecule that cannot escape from the inside?
Oh, that's a good question. So, as you might have noticed from what I talked about, we were interested in putting in biological macromolecules or other macromolecules because the largest hole, which is the square-shaped hole, that's kind of 2 or 3 nanometers across. But so that is a limitation, although, of course, we are not, if you're wanting to use a highly stable cage delivery system, large macromolecules are probably what you want to be delivering, right? Because they would benefit from being inside a stable cage. Otherwise, they would just be digested or broken down in vivo. So whereas small stable molecules, you probably don't necessarily need a cage. So that is a limitation, but it's not really, we don't really see it as much of a limitation in our case.
Okay, and the next question, could you attach protein tags, either his tag, but also some other tags, to the monomers to be able to label the cell?
Yes, you can do that. that's another area I'm not allowed to say too much about, but yes, we can do that. And we're going to be having some papers on that quite soon.
Okay, so let me ask you the last question. I will take advantage of being host of today's meetings. About this light cleavable cages.
Mm-hmm.
I saw that there was a UV mentioned as a light wavelength.
Yes.
This UV is by choice or by pressure from the molecules applied? Because UV is not really the good wavelength in the biology.
No, it's a technical limitation of the wavelength of that UV is quite long. So, it's in some applications, I guess, although this is quite new results, so we need to look into it a bit more. But if you're thinking about how dangerous it would be for a person, it probably, I think it would be okay, but we need to look into this a bit more careful. It's towards the longer wavelength end of the UV spectrum.
Right.
That's a very good point, of course, yes.
Okay, Jonathan, that's the end. Many thanks for accepting the invitation from the Polish Biochemical Society.